Now we're going to look at our body's instruction manual, or the hereditary information that's stored in DNA. DNA stands for deoxyribonucleic acid, and it's basically what holds all of our genetic information that helps our body run, and it contains the genes that we pass to the next generation. Remember that DNA is bundled up into chromosomes, which are found in the nucleus. And we're going to apply this after we learn about DNA and protein synthesis to see how we use DNA to determine family relationships, to determine or solve crimes, and other information we can get from people's DNA. DNA is sometimes called a double helix or a twisted ladder, and that's because of its shape. We basically have the sugars and phosphates along the outside and these bases along the inside, and they kind of like the steps of a ladder. The work of the discovery of not only what DNA was, but its structure is credited to many, many scientists. We're only going to talk about a few. So if you notice, there's these letters here in the middle, A's and T's and G's and C's. Scientists by the name of Edwin Chargaff basically studied all kinds of organisms and found that in any living thing, the amount of A's always equal T's, the amount of G's always equals C's. And so he hypothesized that these must be pairing together. And we actually call this Chargaff's rules. A goes with T and G goes with C, and you'll have tons of time to practice this. If you notice, A and G are kind of bigger, and C and T are smaller. So in order for that step of the ladder to be uniform, you're going to have one big and one little. And so that's why A and T go together and G and C go together. So again, you can kind of see this twisted shape. It's this photograph or this x-ray that really made the difference. If you can imagine, if you've ever had an x-ray, imagine now the microscopic DNA molecule. So if you could take a picture going straight down this helix, Rosalind Franklin and Maurice Wilkins were able to do that. And this is actually what allowed scientists to realize that the bases are across the middle and the sugars and the phosphates circle around in a helical shape around the outside. And so we can see that helix there. The building blocks of DNA are called nucleotides, and they're made up of three parts: the sugar, the deoxyribose, the phosphate, and then one of four nitrogenous bases, A's, T's, G's, or C's. So this kind of gives you a better idea of that. You can see here is a nucleotide. Here are nitrogenous bases. You can see, again, using Chargaff's rules, A's and T's go together, G's and C's. There are these weak hydrogen bonds holding them together. And the sugars and the phosphates from the nucleotides make up the outside or the backbone of the DNA molecule. Two scientists won the Nobel Prize in the mid-1950s for putting together the material and the information from all the various places and coming up with the final specific structure of DNA. And those two people are James Watson and Francis Crick. So here we have our unwound DNA, our double strand. You can see here's a nucleotide here. So let's just start with the most simple. We see here's a phosphate. Here is the 5-carbon sugar, and I only say the 5 because it has 5 sides, if that helps you. That's the deoxyribose sugar. Here are our nitrogenous bases. This is actually what determines what the DNA is saying is the order of these bases. Notice again, you have one big one and one small one, G and C, or A and T, that always go together. We basically get our information. This is what holds the key from these nitrogenous bases. Here's our nucleotide or our building block, our weak hydrogen bonds. And then here are our base pairs, or our complementary bases. And again, as long as you remember A goes with T and G goes with C, if given a strand, we're going to get a lot of practice over the next few days writing complementary strands using Chargaff's rules.